Dieter, we can start. Thank you.
Hello, everybody. This is Dieter Friedl speaking, and I welcome you to our first webinar in this year. It's on biorelevant dissolution testing, and it is a pleasure for me that Maria Verzoni from the University of Athens in Greece will give the presentation. We have the moderator Christos Repas from the University of Athens in Greece, and I will chairing this session. My name is Dieter Friedl, and I'm the chair of the FIP Special Interest Group Regulatory Sciences and Quality, and I'm also the chair of the Special Focus Group Dissolution and in vitro drug release. I'm a pharmacist and chemist from education, and I worked for more than 35 years for the Bayer AG in Germany. I was head of a quality control development department, where I received much experience in dissolution testing and in other analytical techniques in development. And then I was more than 15 years in the global quality assurance, where I was, for example, responsible for Bayer's quality management system, for validation purposes, and last for continuous manufacturing of solid oral dosage forms. And before we start the sessions, I would like to give you a short overview of our focus group and of FIP. And as I said, I'm chairing the Special Interest Group Regulatory Sciences and Quality. And what this group does, it focuses on regulations, analytical technologies required to show the safety and efficacy of drug products, medical devices, including consideration of bioequivalence, biosimilars, and international harmonization, with emphasis on interest and relevance to practitioners in various settings. And the Special Interest Group has 5 focus groups. One is the analytical science, which is headed by Masaru Kato in Japan. One is the dissolution drug release, which is headed by myself. We have the bioclassification biowaiver focus group, headed by Jenny Dressman. Bioavailability bioequivalence by Shinji Yamashita in Japan. Clinical bridging studies, Oliver Hu from Taiwan. And I would like to give you an overview what our focus group dissolution drug release does. It provides a global independent platform for scientific discussions among academia, industry, and regulators on dissolution drug release testing. It reviews and comments on draft guidelines which impact dissolution testing. And the focus group participated in the drafting of the ICH guidance on BCS-based biowaivers, the ICH-M9, and several people were members of the ICH group, and we also provided comments on the draft guidance, and this ICH-M9 guideline and the question and answer paper received step 4 in 2019. The focus group also published papers. One is the FRP guidelines for dissolution testing for solid oral products. One is the report on dissolution in vitro release testing of novel special dosage forms, and we plan a revision for this paper. And we published also a position paper on qualification of pedal and basket dissolution apparatus, and several other papers. Our focus group organized workshops. We had workshops on biorelevant dissolution testing in the United States and Europe, and this is a very prominent topic. And this is also the reason why we have this webinar on biorelevant dissolution testing today. We had a workshop on nanomedicines. We have workshops on dissolution in vitro release testing of novel special dosage forms. And we organize hands-on dissolution workshops in developing countries to improve the drug product quality. During the Corona epidemic, we organized webinars. We had one on qualification of the dissolution apparatus. We had one on the FRP guidelines for dissolution testing of solid oral dosage forms. We had one on vitro in vivo correlation, and we plan other webinars in the near future. And this year, we will have a webinar on semi-solid topicals and suspensions. And when you have topics you are interested, where the group can organize webinars, your proposals are more than welcomed. And to have very independent views on the dissolution topics, I will give you an overview about the members of our focus group. We have members from the regulators from Europe, FDA, United States, and Japan, from industry, from the United States, France, and Germany, from academia, Greece and Romania, from the Pharmacopoeia, EDQM, and USP, and also from WHO. And this is to give you a short overview and as an introduction. And now I will hand over to our moderator, Christos Repas. And I would like to introduce Christos to you. Christos was also a long time a member of the focus group, and he is a professor in pharmaceutics, Department of Pharmacy, National and Kapodistrian University of Athens. His research interests focus on the effects of dosage form characteristics and of gastrointestinal physiology on intraluminal performance of xenobiotics and the development of in vitro tests that are predictive of the intraluminal dosage form and drug performance. He's co-author of more than 160 papers and books, and he is a member of several editorial boards of different journals worldwide. And with this, I would like to hand over to Christos and Maria. Christos.
Yeah, thank you very much, Dieter, for this very kind introduction, and also for the nice summary of the activities of this specific interest group within the FIP. It is really a pleasure for me to moderate this session, and especially because we're going to have a very good speaker and very good scientist, I would say, in this area, in the area of biorelevant dissolution testing. Maria Vergioni, our speaker today, is associate professor in pharmaceutical technology and biopharmaceutics here at the Department of Pharmacy of the National Accompanying University of Athens. She received her bachelor in chemistry in 1994, then her master's in analytical chemistry in 1999, and a PhD in pharmaceutical sciences in 2004 from the same university at Athens. In 2016, she received also her master's of science in medical and pharmaceutical statistics from another university at Athens, public university. And then her research interests focus to oral drug absorption with special interest in special populations, pediatrics and advanced age population, and the physicochemical characterization of the intraluminal environment, as well as the development of in vitro tools that can predict intraluminal drug product performance. She has co-authored more than 100 review papers and research papers. And she's a member of the editorial board of Molecular Pharmaceutics, Journal of Pharmacy and Pharmacology, the Pharmacy Pharmaceutics. She has been involved in several EU-funded training and research programs, including the HPOP, for which she acts as coordinator, PEARL, Orbito, and several others. So with this, I would like to ask Maria to, yes, thank you, share her screen and start her presentation. Thank you, and I wish you nice attendance of this. Enjoy this webinar.
Thank you, Christos, for the kind introduction. I'm trying to sharing with you my PowerPoint, but I cannot find my Zoom screen from the toolbar. Unfortunately, I cannot change this. Sorry for this, I will try again.
If you change the viewing,
I'm trying, but it's not enabled. It's not enabled anymore.
Yeah, it was a few minutes ago. Yeah.
Yes, but now it's gray. So the only thing that I could do now is to change a monitor. Just give me a second, please.
Mm-hmm. Excellent.
I think now it works better.
Yeah. That's fine. Yeah. Thank
you. Good. So I would like to thank you, Christos, for the kind introduction, and also thank the IFE focus group, the Dissolution and Drug Release focus group, and Dieter for the kind invitation to give this webinar to you. I hope that it will be useful for all of you. The title of my presentation is Biorelevant Dissolution Testing. And before starting, I would like to give you the terminology for this and what we expect with when saying biorelevant dissolution testing. An in vitro test that mimics the intraluminal performance of an orally administered drug product and or the active pharmaceutical ingredient, the API, is termed biorelevant or physiologically relevant. Biorelevant data could be used for screening formulations in regard to their intraluminal performance on a relative basis. As an example is the BCS-based bioequivalence approach, or the biology screening methodology that we're going to discuss today, but also to predict the in vivo performance of the drug if this is coupled with physiologically based absorption modeling or physiological based pharmacokinetic modeling techniques. This will be the other example that we're going to see today. On the other hand, a clinical relevant dissolution test confirms or predicts the in vivo performance of the drug, even if it's not performed in non-biorelevant media. So we have two terms, the biorelevant or physiologically relevant conditions and the clinical level dissolution conditions. Today, we're going to discuss mainly the biorelevant dissolution conditions. And the outline of my presentation will be first an introduction to biorelevant media, and then we're going to discuss two examples where biorelevant dissolution testing was performed successfully. And if we get a closer look to gastrointestinal lumen, and based on the experience that we have the last 30 years in the literature data, we can see and conclude that the dynamic environment of GI looming gives complex and variable composition of gastrointestinal contents because of the constant addition and removal of fluids in the different gastrointestinal compartments. As a result, the physicochemical characteristics of the contents can change with the region, but also with the dosing conditions. And when we're going to refer to the dosing conditions today, we're going to refer to the two conditions as defined by the regulatory authorities, the fasted state conditions and the fed state conditions. The fasted state conditions refers to the administration of the API together with a glass of water in fasted healthy adults. On the other hand, the fed state conditions refers to administration of the API half an hour after the administration of a standard meal as proposed by authorities. Usually these meals, as proposed by FDA and EMA, consist of a high-fat, high-calorie meal. The content of the calories is about 1,000, and the meal consists of two fried eggs, French fries, bacon, and also a glass of milk. And the last 20 to 25 years, we were able to identify the physicochemical parameters of gastrointestinal contents after collecting them after the administration of a glass of water or for a standard meal. And we're able today to have the knowledge in relation to the pH, the buffer capacity, the osmolarity, total bile salts or acids that are present in the contents, the short-chain fatty acids, the cholesterol, the phosphatidylocholine, but also the total protein content and the total carbohydrate content. And in this table, firstly, I would like to present you a few of these characteristics regarding the small intestine and also the distal ileum, the cecum, and the ascending colon. And if we go through these parameters very quickly, we can see that there is variability in all cases. That's why values are presented as median and range or mean standard deviation in some times. But what is interesting to see is the change of these parameters along the intestinal tract, starting from the upper small intestine and a value of 6 to 7, going to the distal ileum with a value of 8, and this value remains high even in the cecum and the ascending colon. On the other hand, we have differences in the buffer capacity, which is higher in the ascending colon, maybe because of the presence of short-chain fatty acids, the osmolarity that is bigger in upper small intestine, total bile salts that are present mainly in the upper small intestine, but also present in the ascending colon, and also the cholesterol and phosphatidylocholine content, that the levels of these two compounds are higher in the ascending colon. If now we're going to the fed state, what is of interest is that the pH remains 8 at the distal ileum, as it was the case in the fasted state conditions, and the difference from the upper side, where the pH value is around 5 to 5.5, and from cecum, which returns to 6 at ascending colon. Based on tables like this and the knowledge that we have in relation to the physicochemical characteristics of the gastrointestinal contents, we're able to create different biorelevant media for the fasted state and for the fed state in relation to different parts of the gastrointestinal tract. But the question that arises is what level of complexity is needed for adequate prediction of in vivo behavior. The starting point would be that the closer the test conditions to the GI physiology, the better the chances of predicting product performance are. However, the level of simulation of gastrointestinal physiology required for obtaining information on the luminal performance can vary with the API structure, the type of dosage form, and the type of requested information. About 7 years ago, manuscript has been proposed, a paper has been proposed, in which a different level of simulations have been proposed based exactly on these characteristics. The first is the level 0 simulation of contents. In this level, only the luminal pH is simulated with no consideration of dosing conditions. For this purpose, distilled water or any buffer system with a pH value within the usual GI pH range, for example, the Competia test without enzymes, can be used. This level of simulation can be useful for DCS class 1 or DCS class 3 APIs, and for products like the immediate release tablets without pH-related food effects, or the extended release products that are not affected by buffer capacity. As an example are the osmotic pumps. Also, this media can be used for quality control testing, and when the release is more than 85% within 15 minutes after the initiation of the experiments, in all cases, that means in all pH tested based on the pharmacopoeia. In level 1 media, the distinction is made between fasted and fed state conditions, and in this case, apart from the pH, the buffer capacity is also simulated. This media can be useful for DCS1 or DCS class 3 APIs that have been shown potent pH-related food effects. Also for DCS class 1 or DCS class 3 APIs, but are housed in extended release products when releases is affected by luminal pH and buffer capacity. And if, apart from the luminal pH and the buffer capacity, the osmolarity, and depending on the location and dosing conditions, the presence of biocomponents, dietary lipids, and relevant digestion products is simulated, then we have the level 2 biorelevant media. The level 2 biorelevant media can be useful in order to explore the luminal performance of DCS2 or DCS class 4 APIs. And usually dissolution data that are updated with level 2 biorelevant media are useful for the PBK modeling, and this is an example that we're going to see later. And the biggest latest of simulation is level 3 biorelevant media, where apart from the other components, also the presence of proteins, viscosity, and digestion are also simulated. The viscosity and the digestion can have an impact on dosage for performance. So level 3 biorelevant media should be used only when specific questions need to be answered, and perhaps should be not linked to a specific DCS class. They can be useful in the evaluation of certain type of lipid formulations, like the formulations that are categorized as type 1, type 2, or 3A, based on the lipid formulation classification system, in the evaluation of luminal stability characteristics, but also in the case of modified release formulations, taking also into account the luminal shear stresses. In this table, the composition of biorelevant media in the fasted state are summarized, just to get an idea of how this media can be created. Start for the fasted state, simulated gastric fluid, the FASGF, the medium that simulates the upper intestinal fluids, the FASI version 2, the midgut, the FASI midgut version 2, the medium that simulates the ileal contents, SIF ileum, and the medium that simulates the colonic fluids, the FASCOF. In level 1, when pH and buffer capacity are taken into account, we have to simulate the pH of the contents, but also the buffer capacity using different buffer systems In this case, we are using usually malic acid in combination with tris in the case of Faskov, where we want to increase also the buffer capacity. In the case of ISSGF, hydrochloric solution having pH of 1.6 is used. If in these buffers we add also the appropriate amounts depending on the region of sodium taurocholate, sodium cholate, lecithin, sodium olate, we get then the level 2 biorelevant media. In this media also the osmolarity is adjusted by adding the appropriate amount of sodium chloride. And in this media we can add, depending on the question that we want to answer, pepsin. It's an enzyme, not digestion product, and the pepsin can be added to ISSGF, or if you want to simulate viscosity effects in the lower intestine, we can use, for example, microcrystalline cellulose into the Sif ileum and the Faskov. A similar table we can provide also for the fed state simulation. In this case, the variety of buffer species change depending on the value of pH that we would like to have. And we have to keep in mind that we start from stomach in the fed state, that pH can have a value of 5. Then during digestion period can go to 3. And then we go again to more alkaline values to 6 and 8 in the Sif ileum. So a variety of buffers are proposed depending on the medium that we would like to prepare. And on these buffers, we add the appropriate amount of lipids, bile salts, and also glucose or sodium chloride in order to adjust the osmolarity. And let's now pass to the biorelevant dissolution testing and see two examples. The first example being the evaluation of dissolution in biorelevant media and its impact on the absorption process of low solubility drugs. The example that we're going to see is the SB705498 API. This is an API that it's a weak base. It belongs to class 2. Class based on BCS classification and has a log period 5. The aim is to see if we can predict the different performance of SB tablets versus SB capsules when we have the same dose. And as we can see here from the plasma data, if we can evaluate the higher performance of in terms of plasma levels of tablets versus capsules. So for this purpose, we use the immediate release tablets of 25 milligrams and capsules containing immediate release tablets with lactose as filler per capsule. In both cases, the dose is 400 milligrams, and they were using the dissolution setup that we can discuss here. First of all, in order to evaluate the dissolution rate in the stomach, we're using the level 3 ISSGF using the mini paddle apparatus of Erveka. This means that the initial volume and the volume during the experiment in ISSGF is 250 ml. In order to evaluate the performance of these two products in the upper small intestine, we are using FASIF version 2, again using the mini paddle configuration of Erveka. And again, the volume used is 250 ml. And when we would like to evaluate the performance in the lower intestine, we use a setup that is a two-stage setup. This two-stage ileal colonic dissolution methodology comprises a first stage where we're using 40 ml of Sif ileum, and then after 2 hours of the experiments, we add the appropriate aqueous solution in order to create a Faskov and in a total volume of 200 ml. So we're using this methodology in order to see the performance of the API in the distal ileum and the proximal colon. And this is, in these slides, we can see the dissolution profiles of the tablets in level 3 ISSGF. In all graphs, we can see the performance of tablets in the dissolution experiments with the red squares, and of the capsules with the empty white squares. And in this slide, in this figure, we can see the performance of the API in Sif ileum and in level 2 Faskov. These are the dissolution data and also the solubility data, because with this line we can see the solubility of the API in each medium. So this in vitro data can be used in the PBPK modeling. In the PBPK modeling, apart from the vitro dissolution data, meaning how the solid drug in the stomach is going to dissolve and how it will pass to the small intestine, but also how the solid drug is released in the small intestine and the solution in the small intestine. We need also information from literature in relation to the gastric emptying, but also to the volume of luminal fluids in the different regions. Also, we need in vivo data in relation to the disposition and elimination parameters for the specific API. If we combine all this information, we can use the PBPK modeling using the Stella software, and we can see how we can predict the performance of the two products. Here we have again the individual actual data, and with the red line, we can see the simulated data. So based on these figures, we can see that the average plasma profile after the administration of the 16 tablets was simulated to be higher than that of the 2 capsules, in line with the in vivo data in healthy adults in the fasted state. Another example is the use of biorelevant dissolution testing in the evaluation of differences in early exposure. For this purpose, we are going to discuss the BioJIT system. The BioJIT stands for the Biorelevant Gastrointestinal Transfer System. This system was used in order to evaluate the drug concentration in the upper gastrointestinal lumen after oral administration to fasted healthy adults. And this system is of interest, especially when concentrations could be affected by potential precipitation in the upper gastrointestinal lumen. And in the fasted state, drug precipitation in the upper gastrointestinal lumen could occur after administration of poorly water-soluble weak bases. And this is because of the change of the pH in the fasted state in the stomach, but it's expected to be 1 to 2, to the pH of 6 to approximately 6.8, but it's expected to be in the duodenum. Also, another case are the enabling formulations that have chance for precipitation higher in the upper GI lumen. And I would like to introduce you to the BioJIT system. The BioJIT system is a 3-compartmental model. It uses the gastric compartment, the duodenal compartment, and 1 reservoir compartment. For this setup, we're using the mini vessels of Distec and of Erveca and of Distec. And the initial volume in the gastric compartment is expected to be 250 ml, while in the duodenal compartment, there is a constant volume of 40 ml. These in vitro conditions are based on volume of duodenal contents and also on input and output duodenal rates that were estimated after modeling luminal data of high permeable drugs. Based on this data, the gastric emptying was estimated to be 15 minutes. And the conditions in the duodenal compartment take into account both the transport of a highly permeable API via the epithelium of the upper intestine and also the transit through the upper small intestine. Based on these assumptions and on the data that we were collecting after administration of weak bases to healthy fasted adults, we were able to estimate the flow rates that transfer the volumes from the gastric compartment to the duodenal compartment, but also the volumes at the exit of the duodenal compartment, and adjust also a reservoir compartment in order to comply with the continuous gastrointestinal transfer process in vivo. In the gastric compartment, we started the experiment having level 3 FACE-SGF, while in the duodenal compartment, level 2 FACIF. In the reservoir compartment, a series of phosphate buffer solution containing sodium chloride, bile salt, and lecithin are pumped from here into the duodenal compartment, because the aim is that the composition of simulated duodenal contents, meaning the pH, the buffer capacity, the osmolality, the bile salt and lecithin concentration, remains unaltered during the experiment. In all cases, while the experiment, the paddle are rotating at 75 RPM and all experiments are performed at 37 degrees Celsius. And here is how the biology system looks in real life conditions. As you can see, the biology system requires equipment that is commercially available, like a USB 2 setup and a simple 3-channel pump that can adjust the flow rates throughout the different compartments of the biojet. The pump is controlled by a PC, so it can adjust the flow rates as shown before. And during a biojet experiment, the aim is to collect samples from the exit of the duodenal compartment. And this sample can be divided into some samples. So the total amount per volume can firstly be evaluated. And after the filtration, we can have the apparent concentration of the API in the duodenal compartment. By having in mind the total amount per volume and the concentration, the percentage of solid fraction can be evaluated. That is of interest mainly when solutions are studied in the gastric compartment. Also, part of the filtrate can be transferred to the vial where an excess amount of drug has been pre-weighted, and this can be transferred to a shaking bath for 24 hours at 37 degrees, so the equilibrium solubility can be estimated for each time point that we sample from the duodenal compartment. Based on the concentration and the equilibrium solubility, the degree of supersaturation can be estimated at different time points of duodenal compartment samples. And I'm going to show you two examples. The first example is how biojet can estimate the intraluminal concentrations. And as an example, we're going to use itraconazole products. Itraconazole is a weak base. Lipophilic one has a log peak of 5.7 and a molecular weight of 700 approximately. The dose tested in the biojet system was 200 milligram, and two products of itraconazole were tested. The Sporanox solution, it is a commercially available solution where itraconazole is combined with HP beta cyclodextrin solution, and the Sporanox solid dispersion in HPMC capsules. So when we initiate the experiment in biojet, 20 ml of Sporanox solution are brought to 230 ml of SSGF having a pH of 1.6. On the other hand, when the two capsules are used, then the two capsules are put in a gastric compartment of the biojet experiment, which contains 250 ml of SSGF having a pH value of 1.6. And when we go to the results, we can see in this figure the itraconazole concentration versus the different time points when we use the solution of itraconazole, and the same diagram when we're using the two capsules of itraconazole. With the blue points, we can see the mean plus minus the standard deviation of itraconazole concentration in the contents of the duodenal compartment. And with a continuous line, we can see the mean equilibrium solubility values in the contents of the duodenal compartment. Given that the concentration in the case of the testing of capsules are very low, in this instance, we can see the magnification of this data. And this data, we can compare them with the individual values of itraconazole concentration in the contents of upper small intestine of healthy adults. With the yellow squares, we can see these individual values of itraconazole concentrations, and the gray area is the area that is resulted by having the minimum and the maximum concentration observed in humans. With the dotted line in each graph, we can see the median equilibrium solubility in aspirated samples from the upper small intestine. So on average, we can see that we have a satisfactory prediction of concentrations and also of subsaturation in the upper small intestine, as we can see the huge differences between the two formulations. And the only thing that we can maybe see here is a small overestimation of concentration at early time points, but we have to take into account that there were not many sampling from the healthy adults. It was just 1 or 2 volunteers that gave sums at these time points. But if we take into account that we have exactly the same axis here, we can see that BioJet can predict the different performance of the two products and estimate the intraluminal concentrations at early time points. And the second example is Fibrate. Fibrate was tested at 2 different dose levels, 45 milligram and 160 milligram. And for this purpose, solid dispersion capsules were used. Fibrate is the neutral compound, which has a log P of 5.3. In this case, we can see the apparent fenofibrate concentrations in the duodenal compartment of BioJet after the insertion of 160 milligram tablets and of 45 milligram per tablet. With these bullets, we can see the mean equilibrium solubility values in the contents of the duodenal compartments, which are the same for both those conditions. What we see from these figures are that the contents of the duodenal compartment are supersaturated with fenofibrated in both cases. And given that we would like to estimate the difference in early exposure, in order to see that, we're using plasma data. And from plasma data, we calculated the AUC the first hour after the administration to healthy fasted adults. And we compare the AUC and actually the ratio of AUC of the high dose versus the low dose. And we found that the ratio of AUCs in plasma data was 2.4, but was in line with the BioJet AUC estimated from the data that we took the first 50 minutes after the initiation of the experiment. So differences of AUC values between those were statistically significant for both comparisons and similar to what we saw in vitro. We have tested also other examples with and other products with BioJet, and based on this test and evaluations, we concluded that understanding the impact of gastrointestinal transfer on drug concentrations in the upper intestinal lumen after oral administration of disintegrated solid dose units, suspension, or solution to fasted adults with a glass of water was possible using the BioJet system. Also, BioJet was useful for the evaluation of the precipitated dose fraction in the upper gastrointestinal lumen after oral administration of disintegrated solid dose units containing the drug in solution or of a drug solution to fasted adults with a glass of water. Also, it was useful for the evaluation of the impact of formulation and dose on early exposure after oral administration of disintegrating solid dose units, suspension, or solutions to fasted adults with a glass of water. Finally, it helped us understanding the performance of drug complexes with ion exchange resins in the upper GI lumen after administration to fasted adults with a glass of water. But BioJet was also proved helpful in order to inform the PK modeling for evaluation of the precipitation in the upper small intestine, and this has been done using GastroClass. The precipitation time was estimated by modeling first in the BioJet data, but before that, an effective mean particle size was fit to dissolution data that were generated in level 3 for SSGF in order to estimate the input dissolution kinetics for the system. The resulting particle size for the case of compound B that was tested, compound B was semi-fumarate core crystal, and it was weak base with a log P of 2.9, and was tested as a capsule at a dose of 100 milligram. So the particle size estimate was 100 micrometers. And based on that, the advanced compartmental and transient model a cat through gastroclass, was generated to simulate the condition in BioJIT system. More specifically, here we can see this diagram, the concentration in the duodenal compartment, and with the blue points, we can see the BioJIT data, and with different lines here, we can see what the gastroclass had predicted. And here the default value of precipitation time that gastroclass is using in order to predict the concentration of the duodenal compartment. And here we used higher, longer precipitation times. And we noticed that the longer precipitation times predict the 15 and 45 time points after the initiation of the experiment. With this line, we can see the solubility in FASIF. Based on this value, we were able to predict and allow for a 6 max prediction, at least, within the variability of the clinical data. And with this line, we can see how the prediction was if we're using the default value from gastroclass. And with these two lines, we can see how the Cmax was predicted and the whole dissolution profile here when longer precipitation times were used. So concluding my presentation in relation to in vitro methodology, we think that the availability of in vitro methodology that are based on a detailed knowledge of key aspects of GI physiology and their impact on formulation performance and then to drug release behavior is fundamental to the success and efficiency of oral drug product development. In general, the availability nowadays of robust in vitro tool sets can provide confidence to select the right in vitro tool or tools in order to understand the behavior of each specific oral formulation in the GI tract. Also, the replacement of anical experiments for formulation selection or optimization during early product development with appropriate in vitro tools. This also will help us to have fewer completed surprises in relative bioavailability studies and the reduced frequency of failed formal bioequivalence studies in humans, even to increase the use of in vitro data in submission to support regulatory fields. And all this data that I showed you refers to healthy adults. What comes, we don't know what we expect for advanced age population. And recently the EU funded a project that's called HPOP. HPOP stands for drug absorption in geriatric patients and older people. And the aim is to generate for the first time information of the actual dosage form intake conditions for older people and geriatric patients, and also physiological characteristics of their gastrointestinal tract relevant to drug absorption. This duration of project is 4 years. And it was funding under the Marie Curie Action. It's an IT project, so 11 early stage researchers will work together with university, KU Leuven, University of Greifswald, and National Kapodistina University of Athens, but also close to colleagues from industry like Janssen, UCB, Novartis, Roche, Merck, Bayer, AbbVie, and AstraZeneca in order to fulfill this objective. And we hope that in the next 1 or 2 years, we will have some nice results to share with you. If you would like to have more information about this Marie Curie action, please visit our website or follow us on Twitter or LinkedIn. And with this, I would like to thank my colleagues for the close collaboration all this year, like Professor Christos Repas, but also Alexandros Kourendas, Konstantinos Markopoulos, and Danai Georgaka. I would like to highlight also the help of the Red Cross Hospital and the director of the gastrointestinal unit, Konstantinos Goumas, for his long-standing collaboration in collecting all this data in relation to the characterization of gastrointestinal contents, so to give us the opportunity to have a more rich biorelevant media. Also, Jennifer Dressman from Goethe University always there was a close collaboration with her in relation to the composition and the evaluation and level of simulation of biorelevant media, but also colleagues from FPA partners and Orbito who helped us in order to improve our knowledge in vitro tools, validate and evaluate all these tools. And for sure, I would like to thank you for your attention. And now we'll be happy to take questions.
Thank you, Maria, for a nice presentation on biorelevant dissolution testing. So the program now says that we can start addressing questions that have been submitted through the Q&As section of this webinar. And if you agree, I would like to start with the first question. Question is whether you can discuss most recent advancements in biorelevant dissolution testing, and if there are any improvements compared to what was thought until a few years ago. What's on recently?
Thank you for the question. Actually, I think that the Orbito helped us mature a lot in relation to the in vitro setups, given that the whole work package was dedicated to this action. And these two examples were initiated, that I showed you today, were initiated through the Orbito era. From then, I think that we continue our progress on these tools and to enrich also our knowledge in biorelevant in vitro systems. And the colleagues are working on this, and we're happy to see results nowadays. And the main problem, I think, with the validated in vitro tools is the comparison sometimes with in vivo data. And there is an effort to compare in vitro data with in vivo database in order to evaluate this before going to talk with certainty about the validation of them.
Perhaps if there is any progress in regard to the fed state condition.
The fed state conditions is more tricky, is more difficult to simulate. I think that we're in progress on that. There are different groups working on that. We have some advances also in relation to the biorelevant media, in relation to the fed state conditions. During another ITN, we had enriched our knowledge about the physicochemical characterization of the contents, mainly after the administration of the meal proposed by the regulatory agencies, the FDA meal. And based on this composition, we're trying to improve the composition of gastric and intestinal media, mainly in level 3 simulation, that is more difficult because of the presence of solids and the high viscosity of these contents.
Mm-hmm. Okay, thank you. So the second, I would say, set of questions refers to potential options that may be available to evaluate differences among genders or to evaluate special populations when it comes to biorelevant testing. I mean, both. If there is any guidance, any information how to evaluate gender-related difference on drug absorption, and also in relation to special populations.
To my knowledge, in relation to gender differences, we have not any information that is very clear if there are any differences between the physicochemical, for example, characteristics of the transit time between the two gender, apart maybe for the woman during the menstrual cycle. But I'm not sure if we can discuss if there are differences or not in relation to gender species, because we don't have so much information in vivo, at least. Maybe you can better comment than me on that. In relation to special populations, yes, as I show you, we're trying to do our best, at least for the geriatric population, that is the older people, the geriatric patients, that are the target group of APIs and drug products, to have a better picture of what happened there. There is a gap of knowledge on this area.
Mm-hmm. Mm-hmm. Well, I think there are some old data on the potential extension of gastrointestinal transit time during the follicular phase of the menstrual cycle in women, but these are really old data and they have not been confirmed since then. So it's a long time ago, but I agree with you that, yeah, we lack clear information on potential differences between genders when it comes to intraluminal composition or, yeah, even physiology. Yeah. Okay. Another question relates to the challenges associated with biorelevant dissolution testing and how to potentially overcome these challenges. I would, yeah, you can start addressing this and I can.
Performing an experiment with biorelevant media or biorelevant testing is not always easy. That's why we have proposed also the level of simulation, not to unnecessarily use complex media without be the reason to do that. But in relation to prediction for the R&D performance of a product, I think that the biorelevant testing have been advanced. And if you select the appropriate dissolution testing, depending on the information that you request, you can achieve good results. It's challenging, but I don't know, it's a big discussion around this. Maybe we can start if you want also.
But I think you would agree with me that we should limit our expectations in a way that we should rather look this testing methodology as a screening tool or as an attempt to evaluate what if scenarios primarily. I mean, definitely not to try to predict individual plasma profiles or, you know, evaluate as such the plasma profile, but probably more to make comparisons between different doses, different formulations in a more physiologically relevant way. Would you agree with that? I mean, variability, I mean, has variability of the system been addressed at all today when it comes to biorelevant dissolution testing? I remember that a few years ago, the group from Strathclyde, Wilson and colleagues have proposed a methodology to evaluate, for example, the impact of variations on luminal bile salt levels or, you know, luminal pH values, and have proposed a design of experiment-based procedure to evaluate this, but have not seen much advances the last few years, I think.
I agree with you. This is from this university that they are usually propose a different composition of the intestinal contents, mainly in the first and the third stage, using extremes, the high and the low concentration of each component separately, and they try to evaluate the difference in terms of solubility, because usually they are using this proposed media for solubility purposes, but not more than that, not in the dissolution, for example, only in solubility studies.
Right. I cannot see other questions in the Q&A, or I'm missing. No, there are. Sorry. What would be your recommendation to achieve better prediction? Running conventional dissolution testing or with biorelevant media or BioJIT? That might be. Yeah.
Depends on the type of information that we would like to have. BioJIT have been tested and is useful for evaluation of the early exposure. And I showed also the cases where BioJIT has been applied up to date. but also the biorelevant media, depending on the information that we would like to have, but also the type of API, depending on the DCS classification of the API, but also of the product that we would like to test. We can select the appropriate level of simulation, so to avoid running experiments that require a lot of time of running or expertise in order to run.
And why do you focus so much on weak bases? How about weak acids?
The lipophilic weak bases represent the majority of lipophilic compounds that are now in the market. I think is more than the half, and it's supersaturation, for example, is more likely to occur for weak bases because of the transit of the pH from the stomach to the upper small intestine. The weak acids is the other way around, except if they are administered as salts. But also the aim is not only to focus to the API, but also the drug product. For example, Biorelevant was used for neutral compounds, but for solid dispersion, for enabling products, I mean.
Yeah, but I think if I understand it correctly, weak acids typically dissolve in neutral pH. Once they reach the intestine, they are ionized, most of them at least, and they have limited issues in regard to dissolution of the dose, unlike weak bases, which is exactly because they dissolve in stomach, but have problems maintaining themselves in solution at the neutral pH of the small intestine, the upper small intestine at least, almost. And do you know the material that the paddles are made? It's stainless steel, I think.
Yes, in all cases.
Yeah. That's an interesting question. The next one, how low income and resource limited countries can have this possibility to overcome some of the challenges involved in bioequivalence research. Can you advise is there a way, to reduce expenses and still be able to understand bioequivalent formulations? I guess this touches upon the biowaiver specific interest group, but you can say something.
Yes, I think mainly is for them in relation to biowaiver, but the only thing that I can say is that you can have to use the appropriate apparatus if needed in order to provide the information on yourself before going to bioequivalence study, in order to understand the performance of your product. And depending on the physicochemical characteristics of the API and the type of the product, you have to refer to the appropriate apparatus in order to help you understand the product and how it is expected to work in the system, and maybe couple the information from the in vitro setups with the PBK modeling in order to better understand the performance of your product.
That's the point, I think. I think great hope is exactly this, to feed physiologically based biopharmaceutics modeling platforms with good in vitro data, and hopefully be able to conclude inequivalence or bioequivalence based on the in vitro and the software, instead of going to in vivo studies. But we're not there yet in regard to this. It's going to happen, in my opinion, sometime, but we're not there yet. Yeah.
Well, I would like to add to this question that the ICH issued the ICH M9 guideline. That's the biowaiver guideline based on the bioclassification system, which is applicable to the PCS class 1 and 3 APIs, and very fast dissolving tablets, and this can facilitate to reduce bioequivalence testing and to get biowaivers accepted by the agencies.
Mm-hmm. Yeah, thanks for clarifying, Peter. Indeed. But it has to be BCS class 1 or 3.
Yes.
Under specific prerequisites. Yeah. But yes, indeed. How much does the dissolution testing requirements are different for weak acids? Well, it's not. Yeah, you want to address this?
No, you can go. It's not related to the API characteristics time of dissolution.
It's not the requirement that it's different. It's the fact that you expect, as I briefly mentioned earlier, you expect to have less problems when it comes to weak acids, especially with weak acids that are with pKa's between I would say 3.5 and 6, or yeah, actually greater than 3.5, I would say, because you do not expect major dissolution problems in the small intestine for this compound due to the ionization of these compounds in the relevant luminal pH. But the requirements do not change, do not distinguish between bases and acids. Okay, since we still have few minutes, and I think this time I'm correct that there are not other questions from the audience. Maria, may I ask you, did you mention that with the biojit, transport through the epithelium is taken into consideration? So it has been taken into account for the design of the system, although there is no cell line or absorption, so-called compartment.
Yes.
Compartment.
Mm-hmm.
Yeah.
You're right. Yes, we have based our biojit system on intraluminal data collected after the administration of 2-week bases at 2 different dose levels. Though based on intraluminal concentration of this data, it's more than 300 time points, we're able to simulate the transfer, and this transfer and the volumes also that we created, and also the conditions in the duodenal compartment, take into account both the transport of a high permeable API via the epithelium of the upper small intestine, but also the transit through the upper small intestine. That's why in our biojit setup, we don't have any compartment simulating the absorption, because it's simulated via the different transfer rates we're using in the different time intervals.
Mm-hmm. And there is a question on the chat for furosemide, which is BCS class 4. Right, depending on the dose, yeah, could be. Do we need to conduct a bioequivalence study for the generics, or just do comparative dissolution testing? Yeah, I can take this. It's if it's not class 1 or class 3, and if certain conditions also do not apply, then you cannot implement a biowaiver approach. It used to be the case for some weak acids, BCS class 2 in the past, but I'm not sure if this is still the case, at least from the WHO. But in general, I mean, it has to be highly soluble. So if it's class 4, I don't think so. But Dieter, you can comment further if you like.
When the biowaiver system is only applicable for BCS 1 and 3, that means highly soluble drug substances and their drug products. This is the current status.
Maria, would you like to further comment on that, or?
No, I think it's okay.
Yes. Perhaps one last from me. I mean, you dealt with dissolution in your presentation. Dissolution in the lower intestine. I'm not sure you referred at all to the obviously increased viscosity in the lower intestine. Would you think that this is something to consider in the future when it comes to biorelevant dissolution? Is there any work performed to date in relation to this?
In relation to the dissolution in the lower intestine, and before studying dissolution in the lower intestine, you have first of all to ensure that your API is stable in the colonic contents. So first of all, you have to test the stability of the API in the colon. And once you are okay with this, then you can perform dissolution testing in or target for colon targeting products of the modified release product. In this case, we can propose to have a higher viscosity in this region. And we have already proposed a medium that contain solid parts in order to increase the viscosity. We have done already some experiments with this. We identified the differences in the dissolution in some products that we tested, but these differences are not always reflected finally in the plasma profiles when we included this information to the PBK modeling.
So as I understand, there is more effort to be devoted to this because we do not really know, I mean, how much important this increase would be. I guess it makes sense because it's, I mean, definitely perhaps the solution is maybe retarded, but due to the small amounts that are absorbed per unit time from the lower intestine due to the limited surface area, this might not, impact that much the blood levels. Is that what you're saying?
And also the lower aqueous volume that is present in the ascending colon and the lack of aqueous content in the transversal descending colon. So if you're going to test something, you focus on the ascending colon in terms of dissolution. And also you have to take into account the lower aqueous content of the contents of the ascending colon.
Okay. Well, I think that we are approaching the end. I do not see other questions from the audience.
I don't see any further questions, Christos.
Yeah. So should I give the microphone to you, Dieter, because I think that we have concluded. Yeah. Thank you very much.
Yeah. At the end, I would like to thank you, Maria, very much for the excellent presentation. And I thank you, Christos, for the moderation and all participants for the good questions and discussions. And I would be very happy to have you again in one of our next webinars. The next webinar of our group, the dissolution drug release, will be on semi-solid dosage forms and then on the suspensions. And I would be very happy if you can join those meetings. They will be announced as usual on the FIP webpage under the webinars. Then I would like to thank you for the participation and stay healthy and see you and hear you again in the near future.
Thank you. Bye.
Thank you. Bye-bye.
Bye.